Hey guys, welcome back to Bioinformatics with R. In this video, we're going to actually create our own trees. So in the previous few videos, we worked with manipulating tree files and binding trees and annotating them and comparing them with PCA and things like that. But what if you don't have tree files to work with? You just have sequences and you want to make trees from that. Well, this is the video for you then. We're going to learn how to make tree files. So let's get to work. Alright, so first thing we're going to annotate reconstructing trees from alignments. So we have to first we're going to annotate the packages. So there are 3 packages required for this. So we're going to start with biostrings, which is a package to work with DNA sequences. We're going to open MSA. And we're going to open Fangorn. Okay, so let's quick run these 3. Beautiful. First, we'll load the sequences into a seeks variable. So we're going to say seeks are read AA string set. And we're going to do abc.fa. So as you can see, we have our string set. This is all of our elements. We have the names of the species here. What else we got here worth pointing out? This is an amino acid string, etc. So now let's construct an alignment with the MSA package and Clustal Omega, which you may remember from the multiple alignment lecture. So ALN for align, MSA. So we're going to pull the MSA function from the MSA package. We're going to run it on our seeks, which was our protein alignment files, or protein files. Method equals, and then we'll use the Clustal Omega. So it is now doing the alignment for us. So we've got it aligned. So now, to create a tree, we need to convert the alignment to a phi data object. So we have to kind of reconfigure the way the data is presented so the phylogeny programs can understand how to read it. So we're going to say ALN, and we're going to do as phi dat ALN, and then we have to tell what kind of sequences we're giving it. So we're giving it amino acid sequences. Spell up the D's got to be capitalized as phi dat. There we go. All right, so now if we do class ALN, it should say it's a phi dat. Perfect. In this step, we'll actually make the trees. So we got everything prepared. Now we're going to make the trees. So trees are made from a distance matrix, which can be computed with dist.ml, and ML stands for the method, maximum likelihood. Okay, so dist.mat for distance matrix is dist.ml ALN. Okay, so it just says that it just gives us a printout of what it's doing, I guess. Some functions do, some functions don't. So now we pass the distance matrix to up GMA to make a UPGMA tree. So UPGMA tree is UPGMA of our dist matrix. And we can plot UPGMA tree, and we're going to put a title on the plot. So we're going to say main equals UPGMA, because we're going to do two different plots here. So let's run these two. Okay, so here's our tree. We have Cryophilus, we have Pombe, Japonicus. But the reason I want to show you this is, so now we're going to use, instead of UPGMA, we're going to use a neighbor joining function, which is a different method of constructing for trees. So we say we now can conversely pass the distance. We use the same distance matrix to a neighbor joining function. So we'll call this one NJ, NJ tree for neighbor joining tree, and we'll say NJ of dist mat. Then we'll plot NJ tree, and we'll give it a title too. We'll say main is equal to NJ. So let's run these two. So as you can see, we get two different trees, right? So depending on the method you use and the amount of data that is given to these functions for tree construction, you're going to have two different tree printouts. And so I'll zoom in on the neighbor joining one. So you see here that we have Pombe, Japonicus, and Complicata that are kind of grouped together. If we look at the UPGMA, Complicata, Japonicus, Pombe are more closely related, so it looks different than our previous tree. So you have to determine the method that's best for you, and if you give it more data, longer sequences, etc., it maybe create trees that are more closely related. So lastly, you're going to use the bootstraps.phi dat function to compute bootstrap support for the branches of the tree. The first argument is the object, which is, in our case, is we're going to use align, while the second argument, the function, is the type of alignment. So we're going to use, let's see here. We'll use NJ. Okay, so let's talk about bootstraps quick. So bootstraps are essentially, and this, if you talk to an evolutionary biologist, they might kill me for breaking it down this way, but they're essentially confidence intervals for how sure the clade is placed in the correct position. All right, so first we're going to do fit PML NJ tree ALN. So we're going to run that. Next, bootstraps is bootstrap.phi dat ALN, and our function is going to be equal to function x neighbor joining dist ml and bs equals 100. It's our Bootstrap number. So it says use Bootstrap phi data, and then it says on what data, which is our alignment file, amino acid alignment file. And then it says you need a function, a bootstrapping function. So we wrote our function is to take neighbor joining of the distance matrix and have a bootstrap number of 100. All right, I got an extra parentheses in here somewhere. And 2, comma, bootstrap 100. That's weird, it doesn't look like a bootstrap. Fidat, comma, ALN, comma, fun equals function x, nj, dist, ml of x. There we go. Cannot find bootstrap.fidat. Why can I not find bootstrap.fidat? Because it's bootstrap, not bootstrap. Ah, typos, I tell ya. Oh, still can't find bootstrap.fidat. Bootstrap. Hey, there's something to be said for accurate typing. So this dates me a little bit, but back when I was in high school, like, typing was a class. I don't even know if you have typing anymore because you're all on social media and stuff so much, or on computers so much, but typing was a class, and you type on these programs and it would be like, this is your speed, this is your accuracy. I was a very fast typer, not so much a very accurate typer, so excuse my sloppy typing. Alright, so now let's plot our bootstrap. nj tree bootstraps p equals 10. Okay, so let's, I did run the bootstraps already, so let's just run this. Still can't spell bootstraps. Jeez Louise. Oh, and maybe something should equal 10, like a P. Oh, I saved it as bootstraps. Bootstraps. I'm having such a hard time with that word. It's incredible. All right, so I reran that after I fixed it. Hey, we have a plot. All right, so let's look. So as you can see here, these numbers are the bootstrap confidence interval, so to speak, of like how confident that this tree is joined here, how confident this tree is joined here, so 85 out of 100 times. So it's kind of doing like a random this is coming off the rails, a permutation test. So like 85 out of 100 times, you know, this successfully attached here, 100 out of 100, and 100 out of 100 for those two. So this just gives you, it's a nice way to look at your tree and see if you think it is being constructed accurately. Bootstrapping is a way to determine that. Okay, so you've seen now how to do use alignment files. You can also do this with, instead of amino acids, you could do this with nucleotide sequences as well. So you just read, this would be read NT, and then you'd switch this to nucleotides, etc. Same thing with the alignment that we did previously with the cholesterol omega, but we used protein. So, but this is how you take alignment files, a multiple read alignment, and you can construct trees. So if you have multiple species or strains or ecotypes, if you're doing plants like Arabidopsis, you can create a tree. And then from here, if you wanted to take these files, save it as a new ecotype file, and then go and add the shading, identify a clade, and things like that, you are able to do that. So that's all I got. That's it for this lecture on how to create your own trees from aligned sequences. I hope you enjoyed the lecture, and I'll see you in the next one.